So we're going to take a quick look at learning and memory. Something that came up in 8.4 as well, but we're looking at it again because we're looking at it in terms of what actually happens in the synapse and what are memories, how do we lay down memories, and even how do we recall those memories. Well, an article that you just read, or at least I hope you just read in the Smithsonian, talked about this. And that article was pretty interesting because it talks about how unreliable memories could actually be if something interferes with the synapse. And I hope you read it, I hope you enjoyed it. Let's just go over this passage one more time. So scientists have long known that recording a memory requires adjusting the connections between neurons. Each memory tweaks some tiny subset of the neurons in the brain. The human brain has 100 billion neurons in all, changing the way they communicate. So we're tweaking a tiny subset of the neurons. You might know a little bit more than the average reader about what that could entail. And by tweaking this quote-unquote subset, it's changing the way the neurons communicate. Neurons send messages to one another across narrow gaps called synapses. You definitely know that. A synapse is like a bustling port, complete with machinery for sending and receiving cargo, neurotransmitters, specialized chemicals that convey signals between neurons. So in this analogy, the neurotransmitter is the cargo, and it's being sent and received from port to port, which is the ending of one neuron and the beginning of another. All of the shipping machinery is built from proteins, which are the basic building blocks of the cell. So let's just get rid of that, and we're going to draw a synapse here. And this is a pretty familiar diagram, I hope. You've got the presynaptic neuron and the postsynaptic neuron. So the impulse is being transmitted in this direction. So you know by now that the presynaptic neuron contains synaptic vesicles, and synaptic vesicles contain neurotransmitter, and my neurotransmitter I'm just going to show in red like this. And we also know that there are protein complexes on the postsynaptic membrane. So here's one, a complex does more than one thing. So the two things I'm going to talk about are the fact that it's a receptor, and there is the receptor site right there for these neurotransmitters, and it is also a sodium channel. So once the receptor site is occupied, the channel will be open. Well, the channel's closed right now. Nothing can get through that gate with that closed channel because there's nothing in the receptor. But if I put a neurotransmitter in my receptor site like that, the protein gate will flip open, and I'm just going to try and quickly show that, something like that maybe. And now sodium, which is outside of the postsynaptic neuron, can then move through this sodium channel and depolarize the postsynaptic neuron. So that's really just a recap, you know that. But what you perhaps don't know is that not all neurotransmitters act in that way. The ones that do are called fast-acting. Fast-acting neurotransmitters include things like glutamate. So let's make a note of that, and I think it is worth learning the name of a fast-acting neurotransmitter. And what does that mean? Well, it means that the neurotransmitters that bind on the postsynaptic membrane exert their effects very quickly, but not for long. Fast-acting meaning it's going to exert its effects very quickly, but that doesn't last long, and that's because this neurotransmitter will have an immediate effect. It opens a sodium channel, sodium rushes in, the postsynaptic neuron is depolarized, but it doesn't last long because it's broken down. And that's broken down by an enzyme, a different enzyme for a different neurotransmitter, and there'll be some reuptake of the products back into the presynaptic neuron, and these synaptic vesicles will be recycled and loaded, I suppose, with new neurotransmitter. However, some neurotransmitter will exert its effect in a different way. So now I'm drawing a receptor that is not also a sodium channel, and it has something called a G protein, and this is my G protein right there. I've got another receptor that I already drew, so I'm going to bring that in, and I'm just going to cut out a little notch in that. And this time, when our neurotransmitter binds to the receptor, it's not also a sodium channel, so it can't immediately cause depolarization, but what it can do is cause the G protein to leave and bind here on a sodium channel. So let's just show that happening there, and now I can open my sodium channel. There we go. And now, once again, sodium ions that are outside of the neuron can pass through and depolarize the postsynaptic neuron. But that took a slower amount of time, so it is called slow acting. And an example of a slow acting neurotransmitter is going to be dopamine, which you've heard of, I'm sure. Serotonin is another one. And these are neurotransmitters that are active in the sort of reward center of the brain. We talked about that all the way back in Unit A1. So we associate these neurotransmitters with much more long-lasting effects. We're not talking here about a muscle contraction, a reflex, where it happens instantaneously, and this fast-acting neurotransmitter might be involved in that pathway. This is much more long-term, feeling like the pleasure you get from eating food or having sex or something like that. Now, these have an effect for longer because, I mean, it's slower to start, but it lasts longer because it doesn't matter, there is no neurotransmitter right here that would be broken down. Remember I said that this is going to be broken down and recycled, and it's going to come back into the presynaptic neuron and recycled into neurotransmitters in new synaptic vesicles. Well, there is no neurotransmitter that's bound here. It's the G protein, the second messenger, that is bound, and that's going to stay bound for longer than that would. So it's going to keep the sodium channel open for longer. It's going to keep this pathway being stimulated for longer, and if that does happen to be a pathway involved in the sensation of pleasure, then sure enough you're going to feel pleasure over a longer period of time. So that's how slow-acting neurotransmitters might work. They involve second messengers called G proteins. So a very reasonable question you might have right now is, what does this have to do with learning and memory? Well, these second messengers, these G proteins, also change this entire pathway. They are going to make messages more likely to happen in future. They're going to lay down, they're going to strengthen, I think is probably the best word. They're going to strengthen certain pathways. Important memories may be strengthened quite a lot. Short-term memories might not be strengthened as long as long-term memories. And I want you to just take a second to think, well, what changes could occur that would mean the pathway is strengthened? What do I mean by the word strengthened? What changes might happen here? And what changes might happen here? And if you're thinking, well, maybe you could add some synaptic vesicles, that is a very good suggestion. Yes, slow-acting neurotransmitters may well cause more synaptic vesicles to be laid down in the presynaptic membrane. The other thing that could happen is that maybe some more receptors are laid down in the postsynaptic membrane. I've added a few receptor complexes there. So both of these things, adding synaptic vesicles, adding receptors on the postsynaptic membrane, It's just going to strengthen this pathway. And maybe that is the tweaking. Each memory tweaks some tiny subset of the neurons. That is the tweaking that they talked about in the Smithsonian article. So changing the pathway, strengthening the pathway involves adding synaptic vesicles, maybe adding neurotransmitter to the synaptic vesicles, adding receptors on the postsynaptic membrane. If you've got any questions, you could add them to the question dump or post in one of the discussions. Or you can email me.